So I'm going to be talking about the spore stain and the acid fast stain. So the spore stain, there are two genus of bacteria that will produce a spore, which will be Bacillus and Clostridium. So Bacillus anthracis, Clostridium botulism, or Clostridium tetani. All right, so tetanus. So the reason why these spores are produced by these bacteria is because they, when they are in environments that do not suit them, they will produce these spores. So dry heat, no moisture, no nutrients, they basically are like, I don't want to be here, and they will produce a spore. The spore can survive for many, many years. So that guy that opened up King Tut's tomb, Carter, they say that he potentially died of this because the spores were actually dried, not dried, but in their spore state. So when he had the spores on him and he left, they went back into vegetative cells, which means that they were able to cause an infection. That's how they say he died. I'm not sure. It could be the curse. Who knows? But the other reason why spores can be a problem is because of honey. So children under the age of 1 should not have any honey because of this, because of the spores. So spores are resistant to heat, drying, and UV. All right. So there are three types of cells: the vegetative cell, which is just a cell by itself. So Bacillus and Clostridium are always rod shaped no matter what. Then you have the spore state, and the spore will be produced in the bacteria, and then you'll have the vegetative cell that it basically left. And then you have the germination stage where it goes back to an active cell. So the vegetative cell is the regular cell by itself. It's happy in its environment. When the environment becomes hostile to them, they produce the spore, and then they leave the vegetative cell on the outside because they can survive on their own. That is called sporulation, when it produces a spore. When the bacteria, when the spore is in an environment that it likes again, it will go back into its original form, into a vegetative cell. That's called germination. So with the spore stain, what happens is you need to have a beaker on a hot plate, and the first stain that you'll use is malachite green. So you basically have the slide on top, you add the dye, the bacteria is already smeared on there, and what happens is the hot water bath will drive the malachite green into the spores. So spores will always be green. All right. You rinse with water, and then the safranin will be added. The vegetative cell will be pink. All right. This is the primary stain, the malachite green, because it's the first stain. Safranin is the counter stain because it's the second stain to be used. All right. So next we have the acid fast. So bacteria species, or bacteria that are able to produce, have a waxy substance in their cell wall, are Microbacterium species and Nocardia species. So basically the waxes in there make them hard to penetrate. So when you do this one, you also need a water bath. And then you have the steam coming up with the water, and then you have the slide on top. You will add the carbofusion first, So that will stain any bacteria that are acid fast positive. So that would be the Microbacterium and the Nocardia. So we'll use Microbacterium tuberculosis for this one. So Microbacterium tuberculosis will be stained with carbofusion, and it will be like a pinky color. And you have to use an acid alcohol because it is acid fast. So that's the main difference between acid fast and gram positive. Not gram positive. This turned out horrible. I wish I was in class. When you have the gram stain, you use a regular alcohol rinse. This one is always acid alcohol, thus the name. So then afterwards, you will use methylene blue as a counter stain, and that will stain any bacteria that is not an acid fast bacterium. All right. So acid fast will always be pink. Acid fast negative will always be blue. So acid fast negative bacteria would be any other bacteria, E. coli, Staph aureus, because of this. So yeah, I hope this helps.